Today we will study about the different techniques which are used in prawn culture. So India has remained a competitive supplier of value-added shrimp to the world market because of its low cost of labor and also the scale of economy it has managed to achieve through the export of shrimps. And now India is one of the largest global producers of farm shrimp. In the world, Southeast Asian countries stand first in world shrimp production, and of them India stands second only to China. And taking the global scenario, India is one of the 7th largest producer of shrimp in the world, and in India around 1.8 lakh hectares are under shrimp farming, and of the total global shrimp production of 1.8 to 2 million tons per year, India and China produce a major share. So taking as an example, we can say in 2019 India has exported around 0.665 million tons of shrimp in 2019. Now we will go to the major aspects of shrimp farming. The first one is seed collection, second is spawning and larval rearing, and third is induced breeding. So first, the seed collection. So before going to what is seed, we will just have a look on the reproductive system of male and female prawns. So the male prawns, their sexes are separate in prawns. The male prawns can be identified by the presence of a large copulatory organ which is known as the petesma. So here you can see the petesma which is found between the foremost pair of swimming legs. So this is the petesma. In females, there is a structure known as the thelicum, which is a cup-shaped structure between the last pair of parapods. They provide an anchor site for the male sperm packet because the male releases sperms in the form of sperm packets which are called as spermatophores during mating or copulation. So this thelicum is used for receiving or collecting the sperm packets released by the males. Fertilization of eggs is internal, and the female carrying the fertilized egg mass on her abdomen is called as a buried female. So here you can see a buried female with the egg mass on her abdomen. And then comes the process known as spawning, where the female will shed the fertilized eggs onto the bottom of the water body, and they remain there for a short period until they hatch. Now coming to the various stages. So the egg hatches into the first larval stage, which is called as a nauplius, and it starts swimming up towards the surface. So here you can see the nauplius. This is the nauplius. The nauplia stage goes through several maltings, and it gets larger with each molt, and it ultimately changes to a stage called as the protozoea. The prawn similarly molts and grows through several protozoa stages. So these are the various protozoa stages you can see. The protozoa will then molt to the mysis stage before developing into a post larva. So this is the mysis stage, and the mysis then develops into a post larva, will then become a juvenile, and then it will become the adult, and the life cycle repeats. So this here I have shown the life cycle of Penaeus monodon. So you can see the nauplii, protozoa, mysis, post larva, and the adult here. This is another example as that of the banana prawn, where you can see the zoea stage, the nauplius stage. The eggs, they develop into the nauplius, the zoea, the mysis, post larva, juvenile, and the adult. So this is that of the banana prawn, and you can see juvenile Macrobrachium rosenbergi here. So the first aspect in prawn breeding is seed collection. So seed means a collection of post larvae or juveniles. That is what is called as seed. The sources can be rivers, estuaries, coastal lagoons, etc. So from all these sources we can collect the post larvae and the juveniles. And how are they collected? The gear which is used for seed collection will be a fine meshed bag net, which will be having a receptacle at the cord end. So the net will be fine meshed, and at the end there will be a receptacle in which the larvae will be collected. And the seed has to be sorted, because when you collect seed from the natural water bodies like rivers and estuaries, it will contain seeds of various types of organisms. Not only prawns, it will contain fish seed and other types of organisms from which you have to sort the seed. So, seed sorting is based on certain aspects. First one, the pigmentation. Second one, the shape of the body. And third one, the type of locomotion. So, these are the three criteria which are used for sorting the prawn seed, that is pigmentation of the larva, shape of the body and locomotion. But there is a disadvantage of seed collection from natural water bodies. The first one is there will be insufficiency of seed for stocking in culture systems, because we are going to take these seeds to our culture system. So, we are not sure whether we will be getting sufficient seed by natural collection. Second one is heterogeneity, which I already mentioned. That is, this collection may contain larvae of slow growing shrimp species or predatory fishes. We want the specific type of prawn seed, but that is not possible when we collect seed from natural water bodies. The third one is we have to sort based on pigmentation, shape of body, locomotion, etc., for which we need expertise. We need expert help, which will not always be available. It is very laborious also. And finally, this extensive collection from natural water bodies, it will affect the natural population of prawns. We are collecting many type of larvae, and most of which will die. So, that mortality will affect the natural population. So, these are the various aspects which we discuss on seed collection. Second one is spawning and larval rearing. So, second thing is we can just have brood stock. We can collect mature males and females from natural water bodies, and we can make them spawn under our captivity. So, for this we can collect fully mature or gravid females. As I earlier showed you, a gravid female or a buried female is one which is having fertilized egg mass on her abdomen. So, instead of collecting seed from nature, we can collect the buried females from nature. So, they can be collected from nature or culture ponds, and we usually collect specimens with dark or grey colored egg mass, and they are then released to the spawning tanks, and these spawning tanks should contain clear, fresh and filtered water. So, we directly collect the buried females from natural water bodies and we put them in spawning tanks where they are expected to spawn, and this should contain aerated, fresh filtered water. And the water requirements, the specifications of water also varies according to the species. This Macrobrachium rosenbergi, which is a freshwater species, it will spawn only in slightly brackish water of salinity 12 ppt. Whereas Pinus indicus and Pinus monodon, they breed in sea water of 28 to 35 ppt salinity. So, depending upon the species, the tank which we maintain for spawning should contain water of different specifications. So, what will happen in these spawning tanks? The female will spawn, release the fertilized eggs into water, the eggs hatch out into nauplii. The nauplii will first live on their yolk material, they will transform to protozoa. The protozoa will start feeding on unicellular algae. The protozoa, after molting, it will transform into mysis. Mysis adopts filter feeding on algal cells, and later they become the post larvae. The post larvae can feed on zooplankton, and later they develop into juveniles, which will reach a size of about 20 to 25 millimeter. And now these juveniles can be transferred to stocking ponds, where they will grow to marketable size. So, up to a juvenile, we will maintain them in special tanks, the spawning tanks. But there are many disadvantages of the above methods. First one is insufficiency. You will not get sufficient larvae which you want to take to stocking. Second one is, most of the pinnate prawns attain sexual maturity in natural water bodies. So when you take them to captivity, they may not attain sexual maturity, and availability of buried females from natural water bodies is also not continuous. So these are some of the risks or challenges which the farmers will have to face when they go for natural collection of seed and natural collection of buried females. To overcome this problem, we can go for a technique which is called as induced breeding. That means we are inducing the adults to spawn or breed under our captivity. So how is this done? This technique is called as eye stalk ablation. Eye stalk ablation is a technique in which the eye stalk of female will be cut. So here in this picture you can see the eye stalk of a female being cut. So eye stalk ablation will accelerate the growth and maturation of ovaries in females. So what is the physiology behind this? The eye stalk of a female contains certain endocrine glands. The three main endocrine glands which are present in the female eye stalk are: first is a medulla terminalis X organ. Second is sensory papilla X organ. Third one is a sinus gland. So the three glands are medulla terminalis X organ, sensory papilla X organ, and the sinus gland. The sinus gland, it stores gonadotropic hormones, and these gonadotropic hormones which are stored by the sinus gland, they are responsible for the maturation of gonads. So when the eye stalk is removed, the sinus gland will be removed. And when you do this in the breeding season, in the non-breeding season, so when the eye stalk is removed in the non-breeding season, it will lead to the rapid growth of ovaries. So that is the physiology behind induced breeding in prawns. So when the eye stalk is removed, the sinus gland is also removed along with. And if it is done in the non-breeding season, it will lead to the rapid growth of ovaries, because the hormones produced by the sinus gland, they were regulating the maturation of gonads. But when once they are removed, it will lead to the rapid growth of ovaries. So this is usually done with the help of an apparatus which is called as electrocautatory apparatus. Now usually we remove only one eye stalk, otherwise there will be mortality, because when two eyes are removed, they won't be able to move about, they won't be able to collect food, and so that will lead to mortality. So usually during induced breeding, only one eye stalk will be removed. And we go to the transfer. So once this cauterization is done, these females they will be allowed to maintain in tanks with males. So for one female, we will introduce two acclimatized males. So 1:2 ratio. In 1:2 ratio, we will transfer the females and males into tanks. So they make a brood pair. One female with two males will make a brood pair. So such females and males will be transferred into tanks. And there the gonads will undergo maturation. During this time, we will feed them with nutritious diet, and the water should be aerated and proper pH also should be maintained, during which time the gonads will mature. Around 1 week after this ablation of eye stalk, sexual maturation will occur. Now the females will be transferred into the spawning tanks. So after sexual maturation, there will be development of eggs in ovaries will mature, ova will be released, fertilization will occur, and finally the female will become a buried female. Now these females will be transferred into spawning tanks. Now we will discuss about the types of farms or the culture systems. Shrimp farming is considered to be a very profitable business compared to agriculture and animal husbandry, because the shrimps, they attain fast growth and they reach marketable size in about 6 months. There are 4 methods used for culture in India. First one is extensive, second is intensive, third is semi-intensive, and the fourth one is traditional farming. So first we go to the extensive shrimp culture. This is done in ponds, in large ponds which are of area around 1.5 hectares. But we can only maintain low stocking densities of prawn seed, and we usually use fast growing prawns. So since this is an extensive technique, feeding and pond management is not necessary because we are not investing much money on this. So feeding and pond management is not necessary, and the annual production will be 2 to 3 tons per hectare. So this is called as extensive prawn culture, which is usually done in large ponds with low stocking densities and low management. Second one is intensive culture. Intensive culture, it is done in small and well aerated ponds which are of area 0.03 to 0.1 hectare. So the ponds will be small, they will be well aerated, and there we can maintain a high stocking density of around 5 to 10 lakh of seed per hectare. And since this is an intensive technique, daily water change is needed. We have to feed them with nutrient rich food. The pond management is necessary, liming is required. So we are maintaining the pond well. We can also use circular tanks of surface area up to 2000 meters square with adequate water circulation and drainage facilities, and we need to periodically remove water. Heavy aeration is required, and this is usually containing different types of shelters, artificial substrates, etc. There will be water plants. You can use hollow bricks, plastic pipes, and the substrates offer hiding places for small size shrimps which are just molted. So we will maintain the pond in intensive. We will maintain the pond well in intensive type of prawn culture, where you get an annual production of around 20 to 30 tons per hectare compared to 2 to 3 tons per hectare in extensive farming. So intensive, we spend money and we get money. Third one is semi-intensive. This is done in small ponds of surface area around 0.2 to 0.5 hectare, and the stocking density is lesser than that of intensive culture. It will be 1 to 5 lakh per hectare of seed. The pond management, supplementary feeding, daily water change, etc. will be done, but not as intense as intensive culture. Supplementary feeding is to be done, and we get a production of around 8 to 10 tons per hectare. So this is the third technique. So first is extensive, second is intensive, and third one is semi-intensive. And the fourth one is called as traditional farming. So we will go to the traditional prawn farming practiced in Kerala as well as West Bengal. So first we go to the traditional farming which we use in Kerala. In Kerala, traditional prawn farming is done in rice fields. So this is called as rice prawn rotation culture. This is practiced in low-lying paddy fields. And here what is done is the prawn seeds from brackish water areas and sea will be collected along with the tidal inflow in large stretches of paddy fields. So the seed is collected. Seed collection occurs naturally from the surrounding estuaries or sea water, and the growth of prawns will occur in the paddy fields. And in these paddy fields, we cultivate salt resistant variety of paddy which is called as pokkali. It is a tall variety of rice which grows in flooded waters, and it is tolerant to varying salinities. And after the harvest of this paddy, we will grow prawns in that field. So that is why it is called as rice prawn rotation culture. And this area is called as a Chemminget. It is the rice field or the shallow water body which is used for prawn farming. So the long stemmed salinity tolerant variety of rice which is called pokkali, it is cultivated from June to September, and during harvest most of the straw is left in the field. So you can see residues of paddy field which will become food for prawns. Now the fields after harvesting will be leased out to prawn farmers for a period of 5 months from mid-November to mid-April next year. And prawn larvae coming in from the sea will be attracted by the comparatively lower salinity and the plentiful availability of food in the fields. So they are trapped. And so this occurs during around November to mid-April. So every subsequent high tide during the autumn, the prawn fry will be trapped. That means the prawn larvae will be trapped. So for this, a kerosene oil lamp is hung on the sluice gate to attract prawn fry. And during low tide, the prawns are retained inside the pond by letting the water go out through the sluice gate across a conical bag net having a rectangular frame in the sluice. So it acts as a filter. Only water comes out, prawn larvae will not escape from this field. And harvesting starts from about mid-December until mid-April, and it is done regularly by filtration. So this is considered to be a profitable method. So this is called as the shrimp pokali farming. So here rice and prawn are rotated for culture in this field, and such a field is called as a chemmin kette or chemmin adappu or chemmin wat, and this technique is called as chemmin varth in Kerala. So we will repeat. During June to September or the southwest monsoon season, the rainwater will wash away the salinity of the paddy fields. So during this time we cultivate pokali rice. Harvesting is done during September, and after harvesting long stumps of rice are retained. So during September to mid-November, there will be mild northeast monsoon season where the tidal flow will be prevalent. The fields become brackish. The bunds of the paddy fields are provided with sluice gate. They are kept open during tidal flow. During high tide, there will be inflow of water. During low tide, there will be outflow. During high tide, water with prawn seeds come in. During low tide, bamboo mats or nets are kept at the sluice gate to prevent the escape of prawn seeds. Only water will go out, and the process is repeated several times. And the prawns grow till April to May. The harvesting is done during morning or evening, a few days before or after the full moon day. And what we use is, water is filtered using a large conical bag net, during which the mature prawns will be collected at the chord end of the net or the posterior end of the net, and they are separated. So this is usually done in Thrissur, Ernakulam, Alappuzha and Kottayam districts of Kerala, and it covers around 12,000 hectares of area. And the production rate is 200 kg per hectare per crop from the pokali fields. And the stocking density which we can maintain is 3,000 to 5,000 fry per hectare. And the advantage is there is no need of a supplementary feeding, because the prawns will subsist on the stumps of paddy after harvesting. And there are some techniques for improvement, that is, we can manure the fields, and we can do controlled selection of the fry, and afterwards if we culture, the technique will be more profitable. So that is the technique of prawn farming, the traditional method of prawn farming used in Kerala, done in Kerala, which is called as chemmin varth, that is rice prawn rotation culture, or the cultivation of prawns in rice fields along with pokali rice. So you can grow only pokali rice here, because pokali is the only salt resistant variety. Now we will go to the prawn culture in berries, that is usually practiced in West Bengal. The low lying areas of Sundarban region, you can see this type of culture. And berries are embanked brackish water enclosures which are used for aquaculture. So you can see the berries here, they are deeper than pokali fields, and these berries, they are surrounded by earthen dikes which are constructed by borrowing earth from the trenches excavated inside the berries near the toe line of the dike. So you can see here the dikes you can see here. The dikes around the berries can be seen. So these are the dikes around the berry. So they form the boundaries, and they are constructed by taking earth from the trenches which are excavated for building the berry. And as it was already mentioned, they are deeper and larger in extent than the Pokali fields of Kerala. And on these buns, the boundaries which are seen around the berry, there will be sluice gates, and the technique is the same. That is, during high tide the water will enter into the field, but at low tide the water will flow back through the sluices, and during that time the sluices will be covered with screen made up of bamboo slates. So during high tide, when the water comes in, there will be coming in of prawn larvae, fish larvae, etc. During low tide, when the water goes out, because of the closing of the sluice gates, or because of the presence of nets at the sluice gates, the prawn larvae or the fish larvae are prevented from escaping. So they will be held up in the berry. Now, the culture period. The culture begins in January to February, during which time the berries are stocked with fry of fish as well as prawn. Occasional harvesting can be done from May to June, and the main harvesting is done from September to November. So this is the traditional culture which is practiced in West Bengal. The berries constitute one of the most important fishery resources in the estuarine wetlands of the upper and lower Sundarbans of West Bengal, which covers around 33,000 hectare area, and the berry fishery is locally known in Bengal as Basa Banda land. The berries are of seasonal as well as perennial type. Seasonal means used for only a particular season, perennial the year around. The seasonal berries dry up during November to December, during which time they are exposed to sun for a month till the next season. Both fish and paddy are raised in the same unit in the sequential manner, sometimes in conjunction, so sometimes together. These mainly exist in low and medium salinity zone, because rice is also to be grown there. When rice is grown along with prawn, the rice we have to select should be, the farm we have to select should be of low or medium salinity. The perennial berries exist in high saline zones in the southern part of the Sundarbans. The fish and shrimp will be raised almost throughout the year. The bed of these berries is not generally dried, and paddy is not grown here because of high salinity. So in perennial ponds, perennial berries we don't cultivate rice, whereas in seasonal berries we can cultivate rice, just like we do in Pokali farming in Kerala. But there are some environmental problems in the berry system. The flood and rot during culture operation may inhibit the culture. Silting up of feeder canals and creeks may occur. That will also affect the culture. Pollution of water sources through the discharge of sewage effluent. Excessive growth of filamentous algae and weeds will cause problem of oxygen depletion, because they decay in summer and they lead to the production of noxious gases. So we go to the general culture management practices when we do culture in ponds. The production pond should be effectively managed for increasing the production. Liming should be done at the rate of 1000 kg per hectare, depending upon the nature of the pond bottom. Liming is used for removing the parasites and to maintain the alkalinity. The water in the culture pond should have a pH of 7 to 8.5. Manuring is also essential to increase the productivity of the pond. The juvenile shrimps are stocked in culture ponds after acclimatization. The stocking density varies according to the species. It is 30,000 per hectare for Macrobacchium and Pinus monodon, whereas it will be higher for Pinus indicus, because it is a small sized shrimp, that is 50,000. For Macrobrachium and Pinus monodon it is 30,000 per hectare, whereas for Pinus indicus it is 50,000, higher stocking density because it is a smaller one. Mixing of different species and size groups is not advisable in shrimp farming, because they are cannibals. They will mutually eat. So we do not mix the different species. Supplementary feeding, if you are doing in shrimp farming, You can use feed materials such as trash fish, clam meat, mussel meat, egg, rice bran, oil cake, tapioca powder, fish meal, etc. along with the quality formulated pelleted dry feed. So, as a rule, the shrimps grow well with a highly proteinaceous food which is having a protein content of above 40%. The feeding is performed up to 5 times daily. In small ponds, the feed may be spread all over the water. It is better to place the feed in a feeding tray and immerse it at different localities in the pond so as to avoid the decaying of the pond water because of spread feed. Shrimps can be harvested after 5 to 6 months. So, these are the general practices which we do when we farm shrimps in ponds. Then harvesting. Most of the prawns are active during the full moon and the new moon days and also shortly after sunset and just before sunrise. So, we usually regard a few days after full moon as the best time for harvesting. When they are not very active, we can easily catch them. Then, in non-drainable ponds, cast nets and seine nets are used for harvesting. So, they are the two types of nests used for harvesting. In drainable ponds, a bag net will be placed at the water outlet for collecting the prawns. The prawns trapped in the pond mud are hand picked, cleaned and marketed. So, when the pond is non-drainable, you can use a cast net or a seine net. Whereas, if it is a drainable pond, at the end of the drain, you can place a bag net in which the prawn will be collected. Otherwise, you have to go for a cast net or a seine net, after which they will be marketed. So, this finishes the techniques used in prawn farming.